Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this news briefing from the ACS Fall 2019 National Meeting in San Diego. We're joined today by Dr. John Termini from City of Hope. He's studying how diabetes can increase cancer risk. Dr. Termini.
Thank you, Sophie. So it's been known for a number of years that having diabetes, mostly type 2 diabetes, but now it looks as if type 1 may be involved as well, increases the risk for specific cancers that would be including endometrial, breast, ovary, colon, pancreas, and liver. And the association has actually become even stronger as we've done more data, collected more data from trials, and analyzed data from very large sets over the years. Now it seems as if there's actually sex differences that have been determined. It seems as if women have 6% higher overall risk of cancer as a result of having type 2 diabetes. So the reasons for this traditionally have been primarily endocrinological in nature. Hormone levels are increased as a result of obesity, which is a risk factor for cancer. Aromatase, estrogen increases, and also insulin levels, which are high in type 2 diabetes. People with diabetes have excess levels of circulating insulin, and insulin is unable to carry glucose into the cells to perform its metabolic function, and the pancreas respond by producing excess insulin. And this hyperinsulinemia is a mitogenic thing which causes cell proliferation. So these are the common ideas that have been circulating around. There's some evidence for those things, and no doubt they play some role. But because we think of cancer as a disease of DNA, it's a DNA damage, and rearrangement of DNA mutations cause our major driver of cancer. We wanted to look at ways in which complications that occur as a result, metabolic disturbances that occur as a result of diabetes may actually impact DNA and give rise to this, what's termed genome instability or DNA damage, and maybe drive as a driving force in cancer. So, and actually at City of Hope, there's a very long history of diabetes research. It's known primarily as a cancer institute, but it's basically, it's also there was a landmark discovery. The first synthetic insulin was created there in the late 70s. So there's a large group of individual investigators who are interested in that. And we were turned on to this idea by a fellow named Dr. Sam Rabar, who was a famous man in his own right, who discovered the relationship between diabetes and hemoglobin A1C. And he came to us one day, he said, you know, you study DNA and damage and mutations. Did you know that sugars and carbohydrates can actually react with DNA and cause products that are mutagenic? And we didn't know much about it. So we started studying this. And even though there was a lot known from basically the food literature, for where people study the reactions of sugar with food, there was really no indication whether this had any type of biological significance. So we developed some methods, analytical methods to measure this, and we're the first lab to report DNA damage caused by glucose, by hyperglycemia in humans. And subsequent to that, we've been studying a lot of the chemistry of these adducts, how they may be replicated, what kind of mutations they may cause in DNA, and also the fact that they can also cause DNA strand breaks, which is another catastrophic event in DNA that's also a significant driver of cancer. So we studied those in mice models of diabetes, and recently we've completed a clinical study where we showed that these DNA damage adducts are also elevated in people. And most recently, what we presented at the meeting yesterday was data that shows that the repair of DNA adducts is also decreased metabolically in patients with diabetes. And this is a consequence of elevated glucose, and it shows that there's the relationship between metabolism, altered metabolism in diabetes, and how that can impact basic biochemical pathways that are important for maintaining the integrity of DNA. And we believe that these things can enhance cancer risk. In addition to lifestyle changes that people can affect to maybe reduce their risk in terms of controlling their obesity, exercising more, the kinds of things that people hear about all the time, there may be pharmacological approaches that may actually help ameliorate or improve the outcome of these biochemical pathways, which are perturbed as a result of high glucose, and maintain their DNA stability over longer periods of time. So that's basically what we've been working with.
All right, thank you very much. We can now open the floor to questions. If you have a question, please state your name and affiliation.
Laura Cassidy with ACS. So are there existing drugs that can boost DNA repair mechanisms?
They've been looking at that in terms of chemotherapeutic drugs, but we're actually looking at drugs that boost the metabolic parameters that are impaired. So there's things called prolyl hydroxylases, which govern the transcription of genes which take care of DNA repair, and there's also signaling pathways which are impeded as a result of hyperinsulinemia, which also affect the DNA repair pathways. So indirectly, they would affect DNA repair, but they're not actually targeting the DNA repair machinery directly.
I see. And are you testing any of these in animals to see if you can reduce the cancer
We
risk?
will. We're doing it in cell models currently, and we've seen interesting and encouraging results in cell models, and we're looking not just at expression of repair genes and proteins, but also we have an assay to measure repair of the DNA damage that's caused by the elevated glucose. So specifically, it's a specific kind of DNA repair for that type of DNA damage.
Katie, excuse me, Katie Cottingham, American Chemical Society. So you mentioned that there's some medications that already exist that could possibly help this situation. Are you also working on developing new medications for this?
Not exactly. The reason we're not is because there's already a number of drugs available that target some of these enzymes, and some of them actually are in clinical trials for other reasons, like for example, for targeting renal cancer. They use EPO, drugs that stimulate production of EPO, actually also impact this pathway because the transcription factors involved are the same. So there's a possibility that and that's where our first shot would be trying out things that already exist and looking at those effects. And if, as we need to, we have a pretty strong chemistry group as well who are always interested in new targets, and that's something down in the future.
That's good.
Laura Cassidy with ACS. I believe in the press release you said Metformin was one possible drug that could help with this DNA repair problem. And I know that's commonly given to diabetics already, correct? Is there any data to show that diabetics who are taking Metformin have a lower cancer risk than other
It's emerging,
diabetics?
yes. There is actually some data now that's showing that, and it's another reason. Metformin's a great drug. Actually, Metformin's shown to stimulate DNA repair. There's one or two scattered reports in literature. We're looking at other things where they showed that Metformin can actually stimulate nucleotide excision repair, which is a type of enzymes in that pathway. So we actually have a study using mice we've been treating with Metformin, and they're diabetic, induced by a high-fat diet, and we're actually looking at repair activity from specific tissues of that mouse and see whether Metformin, and insulin as well. Insulin also has been shown to stimulate DNA repair. But by themselves, that may not be enough, so we're looking at other things to affect the metabolic pathways that are impacted.
Bill Buslik, ACS. Do I understand this correctly? The DNA damage is actually adduct formation, and then that gets excised, and then you measure the free floating adducts in blood.
Yes, so the adducts themselves are they're not direct glucose adducts like HbA1c. They're glycoxidation products and other byproducts of glucose metabolism. So they're alpha oxoaldehydes Methylglyoxal is known to be elevated significantly, 6-fold in patients with diabetes. So we actually initially measured these in urine. So they're excreted into the urine systemically as a result of probably a combination of DNA repair, where they're excised and excreted, and just cell turnover. But we've also measured it in tissues, especially in tissues that have been identified as being more prone to develop cancer in a mouse model. So we can measure, in that case, yes, we extract DNA, hydrolyze it, and we can measure that as well. So in tissue, and also in plasma, but we prefer measuring it in urine. It's a more simple and more direct assay.
Now, you mentioned the metformin. In fact, Laura was asking about the metformin. Did you ever actually measure people? There are probably hundreds of thousands of people out there that are taking metformin for type 2 diabetes. And did you have any data to indicate that it's a reduction of the adduct formation in that particular instance, or are you just getting ready to do that?
We're not there yet, but the mechanistic information, I mean, metformin does a lot of different things. It scavenges aldehydes, it affects the AMPK phosphorylation, and also it appears to increase levels of alpha-ketoglutarate, which is one of the metabolites that affect the transcription of NER genes, and we were surprised to see that. To learn that, we didn't see that, we learned that, and that's another mechanism whereby it can affect the repair. So yeah, metformin does a lot of things, and we haven't really, we're starting that, we haven't really done much with it at the moment.
Thank you.
Hi, Zen Vuong with City of Hope. So people have discussed the link between diabetes and cancer for a while now. Can you tell me how your research, how the finding is new and different from what is known?
Well, there really has never been a DNA damage hypothesis introduced as to how that risk is incurred. And I think that's a novel finding, and I think it also suggests other ways of maybe, for folks who are unable to maybe correct their lifestyle or to control their diabetes. Glycemic control is very difficult in the populations. I mean, studies that have been done in Joslin and in other places where they can control people very strictly, you can do that, and that's where most of our information comes from. So what do you do with those folks who are unable to do that? And how can you help them reduce their cancer risk? And I think you can't really change their hormone levels that easily, especially if they're obese. The adipose tissue is actually producing a lot of the hormones that are mitogenic and are carcinogenic. But I think by able to correct this DNA repair defect separate from the other metabolic disturbances, I think provides another way to get that risk reduction. And so I think the DNA damage hypothesis, people have known that carbohydrates react with DNA, but how it specifically relates to enhanced cancer risk is a new finding, I think.
Any further questions? If not, thank you very much. The archived version of this session will soon be posted at bit.ly/ACS 2019 San Diego. Please join us for our next press conference tomorrow at 9 a.m. Thank you very much.